Hey there, everybody. Mr. Larson here. So now that we've talked about what DNA is sort of used for and how it's replicated, we're now going to dive into this process called transcription. So now we're going to figure out how DNA is used to create different molecules. So this is through a process called, or the start of it is basically the process called transcription. So now what happens with the DNA is that it is copied to create what is called RNA. So RNA is ribonucleic acid, just like DNA is a nucleic acid, so is RNA, which contains the coded information for making proteins. So DNA overall creates proteins. And these proteins are used for very different things throughout our body, but DNA is the template that creates these proteins. So transcription is the process where messenger RNA, so a specific type of RNA, is basically created from a DNA molecule. And so this is all done through an enzyme called RNA polymerase. So transcription is basically taking DNA and turning it into RNA. So how is that done? We will see here. So RNA polymerase comes in to the DNA molecule. It unzips it just like how a DNA polymerase does the same thing. So polymerase, unzip. And the RNA polymerase brings in different RNA bases. So these different RNA bases basically come in, attach to the DNA, and create their own specific strand, basically copying the DNA. So instead of replicating the DNA, we are now creating a different type of nucleic acid, which will be later used to create proteins. So now that the messenger RNA has been created through RNA polymerase, basically we need to figure out what RNA really is. So RNA is ribonucleic acid. It is different in DNA due to three different things. It is a ribose, so it's not a deoxyribose, so the type of sugar is different. It is single-stranded, so you don't have the two bases coming together. It is only one strand. It contains uracil instead of thymine. So basically what I mean by that is wherever there is a thymine in a DNA, you're going to replace it with a U or uracil. So there is no such thing as thymine in RNA. It is only uracil. So for RNA, the adenine, the A, is their best friend is uracil. So mRNA also means messenger RNA, tRNA is transfer RNA, and rRNA is ribosome RNA. Each of these is used in the process of translation and transcription, which we'll talk about translation here in a little bit. So yeah, RNA, the main idea to think about, the main difference is this right here. It contains uracil instead of thymine. So that is the real large difference that RNA has compared to DNA. Other than that, it's very similar. So this is a concept map just showing what RNA basically does. So it can either be one of three things. You can either have it messenger RNA, ribosomal RNA, or transfer RNA. So let's look at messenger RNA first. So it's also called mRNA. Basically what it does, it carries instructions from the DNA to the ribosome. So the ribosome is what basically, we've talked about this in our cell digital lesson. Ribosomes basically create proteins. So basically the messenger RNA carries the instructions from the DNA and brings it over to the ribosome. A different kind of RNA is ribosomal RNA. So basically what it does is it combines the proteins to make up ribosomes. So just like the name, ribosomal RNA creates ribosomes. Kind of easy to figure that one out. So transfer RNA. So basically what transfer RNA does is it brings amino acids to ribosomes. So we'll dive more into what amino acids are and how they sort of function in the whole idea of RNA, but the transfer RNA is what brings those amino acids from around the cell and brings it to a ribosome to create these proteins. So we're going to hear in a little bit, this next video, we're going to be talking about translation, what that means, and how amino acids are actually used in to create proteins. So I hope this helps out, and I will see you guys soon.